Hey scientists, so greetings from my old alma mater, from Colorado State University. So without further ado, let's start talking. Other genetics, beyond the basics, part 1. So let's do a little review. So we have the single genes with the two alleles. So many human traits, as you know, are controlled by the single genes with two alleles. You've got a dominant, you've got a recessive. So right now I'm kind of talking this basic Punnett square, right? So you got the dominant capital, you got the recessive lowercase. So for example, the widow's peak allele is dominant over the straight hairline. So with a heterozygous parents, you're going to get a 75% probability of the widow's peak. So like I said, we're talking kind of this Punnett square over here. Another example would be dimples. Dimples are dominant, so they are going to mask a recessive allele. All right, so now we're talking, there are two different types of dominance. The first is known as codominance. This is when the alleles are neither totally dominant or recessive, so they both show up and they're both dominant. So neither allele is masked. So the way this is shown with the letters are capital with a superscript. So what that looks like is, as you can see right here, we've got F. Again, I'm talking about this this Punnett square right here. So we're talking, I'm going roosters. Yeah, roosters on that. So the roosters, we're talking feathers. So F represents the feathers. Okay, and then in this case, as I said, the letters are in superscript. So a capital B would be for black, and then again here we have the capital F for feathers, and the codominant allele would be W for white feathers. So as you can see, this is an example of being codominant, where we've got two dominants kind of sharing space on the allele. So we've got one for black, one for white. That is an example of codominance. Come on. All right. So the next is incomplete dominance. So what incomplete dominance is that's when there's a type of dominance. It's a type of codominance, but the traits blend. So if you look over here at our, I don't know, maybe carnations. We'll go carnations. And I would like red. So all right. So we've got dominant red, and we've got dominant white. All right. So again, here's our Punnett that we're setting up. Bringing them together right here. So what happens is, excuse me, what we've got the red and the white. So they are going to blend right here. So again, they're not, it's not like the codominance right up here where they both express the same way. They're going to blend. So obviously if you get a red and a white, you're going to kind of get a pinkish kind of carnation right there. So the traits blend together. They don't just sort of stand separately like the codominance. With incomplete dominance, they kind of blend, and in this case, you get a completely different color of flower. All right, multiple alleles. All right, so the deal on this, there are some traits that are controlled by a single gene, but that single gene might have more than two alleles. So in this case, and remember, the genes make up the alleles. All right. So with multiple alleles, there are 3 or more forms of a gene that can code for a single trait. All right. So an example for that might be with chinchillas. All right. So chinchillas, we're talking fur color here. So with chinchillas, they can blend, or excuse me, they can code for black, white, gray, or tan. All right, so those are all on the single gene. Okay, in this case, the trait right here would be fur color. Okay, the color of fur. But what's happening is you've got kind of a lot of things living on there. You've got a black allele, you've got a white allele, you've got a tan allele, and you've got a gray allele, all on this single gene. So you've got multiple alleles on one gene. But remember, you can still only carry 2 alleles, because yes, the chromosomes have to exist, and they have to come together in pairs. All right, so as I said, each chromosome in pair, each of the chromosomes in pair carries only one allele for each gene. But again, you can have different forms of that gene for a single trait. A great example of this is a great example of codominance with multiple alleles is blood cells. All right, so basically blood is controlled by multiple alleles. There are 4 types that exist. You've got A, you've got B, got to use red for this. So you've got A, you've got B, you've got AB, and then you've got O. So those are the multiple alleles we're talking about, 4 total. So these three alleles control inheritance of the blood type. So I know what you're thinking. You said there were 4 up here. This one right here, O, is actually the absence of A and B, is the absence of the A and B allele. So you've got three alleles that control inheritance of the blood type. So for instance, we've got I, A, so we've got dominant A type, dominant B type, and recessive. Okay, so we've got the blood types A and B. Okay, and this is the way I'm expressing them this way. So IA, capital I, obviously because it's dominant, and capital IB. And remember, since these are capitalized too, these are also both dominant. So as we said on the last slide, these bad boys are codominant. They both hang. Neither one masks the other, and they do not blend. So that's why, and right up here, as I said, this type right here is recessive. So blood type O is written I, and as it is a lowercase letter, it is recessive. So like I said, technically, type O blood is just the absence of A and B. So if A and B are not present, you get type O. Okay, so let's just kind of run like some really quick numbers here. So if you've got, let's see, if person inherits 1A or IA, from one parent, and IB from one parent, from another parent, then you are going to have a codominant with multiple alleles, which means you are going to get IA and IB, or more familiarly, you're going to have type AB blood. All right, so you may be wondering, how do you get type O blood? Well, let me tell you. So in order to have type O blood, you must inherit 2 recessive alleles. Okay, so type O blood equals 2 recessive alleles. All right, so we're going to test your knowledge, whip out your ISN. Again, you got those because you're already taking notes. On the left side of your ISN, using Punnett squares, I need you to calculate all the possible genotypes for the following P1 combinations. Okay, so parent 1 combinations. So we've got BO, and we've got AB. We've got AO, and we've got AA, AO, and BO. So there are going to be a total. So these will all be right here. You are going to need to do 3 Punnett squares. Okay? Now, remember that the different phenotypes, these are sort of the different physical characteristics. Remember, these are the genetic characteristics, and these are how the genes express as phenotypes. The different phenotypes are A, B, AB, and O. So go ahead and run those Punnets, and we'll go over them in class. That's about it. Other genetics, beyond the basics, part 1. So in other beyond the genetics, beyond the basics, part 2, we will go even more beyond the basics. All right, so I'm out from goodbye from CSU. We'll talk to you later.